Our next presentation is by Dr. Davin Henderson. Dr. Henderson received his PhD from the University of Minnesota in biochemistry, molecular biology, and biophysics. He's currently an assistant professor at the Prion Research Center at Colorado State University, and he works to understand the pathogenesis and replication potential of chronic wasting disease prions. He's got 7 years of experience working with RT-Quic assay and over 17 years of experience in protein biochemistry. He's author of a bunch of manuscripts working with detection of CWD in excreta live animals, and he's here to give us a little more of the technical side of what we have to take into consideration when we're trying to figure this whole thing out. So please welcome Dr. Henderson.
Thank you very much for inviting me. I really appreciate the opportunity to talk to you about this. So I'm glad I'm not the only biochemist here. We had Rodrigo earlier, which makes me feel a little bit more comfortable. So I was asked to try and give an overview of the types of assays that we could use to increase sensitivity for live animal testing. And those two assays are essentially RT-Quic and serial protein misfolding amplification, which you heard about earlier. So I'm going to kind of give an overview of those two tests, but I like to make one disclosure. I am in the process of developing a commercial interest for CWD testing that has not come to fruition yet. So the basic USDA guideline for testing a positive animal for CWD is an immunohistochemistry test in either the obex or the retropharyngeal lymph node. And I wanted to mention this because the retropharyngeal lymph node from our testing and from amplification testing is pretty much the earliest tissue we know of that's positive in the disease. The tonsil in one paper was using RT-Quic was detected earlier. So essentially, lymphoid tissue of the head and neck is positive before the brain. And that's when we were talking about animals that are lymph node positive versus lymph node and brain positive. The lymph node only animals are earlier stage of disease. So catching those is kind of what we're really looking for with an antemortem test, along with the animals that are in these later stages of disease. So there currently is no live animal test available for chronic wasting disease. I can look for my pointer, there it is. And the rectal biopsy test has been suggested as a potential sample for that, but it's not approved for routine monitoring. And the other thing I wanted to mention is that no test is definitively negative for CWD. It's just reported as not detected. And that's important because even the retropharyngeal lymph node is not positive until after a certain point of disease. So we could inoculate a deer and then wait a certain amount of time, and that retropharyngeal lymph node can be negative in very early CWD. And that animal could eventually develop CWD, but it would have been undetected. So I also wanted to mention a couple of the things we talk about when I'm mentioning asymptomatic phase versus symptomatic phase. So the asymptomatic phase is characterized by positivity typically only in the lymphoid region, and it's prior to neuroinvasion. So the obex would be negative by IHC and potentially even negative by amplification testing. And this phase can be longer in resistant genotypes. I'm going to get into that a little bit later. But it was mentioned that the 96G and 96S animals have different disease courses, and they could potentially be longer. And there are other resistant genotypes which could potentially even be longer than that. And it's also one thing we've realized recently is that in experimental dosing, if you use a lower dose, it can also take longer for the disease to manifest. And that's true in nature as well, right? You could have one animal that contacts CWD from a carcass at a very, very high level of exposure, and you could have another animal that could be exposed through contaminated ground or something, which would be an extremely low dose. So that also affects the length of disease course, and that's something I don't think we really spend enough time researching is how that dosage affects the disease course. In the symptomatic phase, we're all fairly familiar with it. Positivity is both in the obex and the lymph nodes, and this is typically easily detected with antemortem testing in remote tissue and in blood and some of the other studies. So the essential difference in the tests I'm going to talk to you today about are a direct detection test and an amplification test. So a direct detection test is detecting the actual prion in the sample. So typically it's based on protease resistance, digest away the normal PRPC and detect the PRPSC. So, and beyond that detection level, we can then potentially rely on amyloid amplification techniques like RT-Quic and PMCA, which could take that small amount of prion, which is undetectable by conventional means, and amplify it to detectable level, right? And these methods can be typically about 10,000 more fold sensitive in the least sensitive versions, and then up to billion sensitive in PMCA. So the detection level for these two types of tests can detect significantly less prions in a sample. So we already have a live animal test for CWD. Why do I want to tell you a bit about how to use amplification tests to improve this? We've been doing this for quite a few years. Not me personally, but labs at Colorado State and the USDA and other agencies have been using rectal biopsy tests and examining them by immunohistochemistry, tonsil tests here in Texas by immunohistochemistry, and these tests work pretty well. The diagnostic sensitivity in GG deer was 76% in this particular study, and it was actually 100% in animals that were in the later phase of disease. However, it's going to miss samples that are in the earlier stage of disease, and that's depicted here where 42% of GS animals and only 36% of animals in early clinical stage of disease with OBEX grades of 0. So these are the numbers we'd like to increase. So theoretically, if you want to test a population with a live animal test, you want to catch as many as possible. And hopefully the live animal or the amplification techniques can help with that. And the two that have been predominantly used, like I said, were RT-Quic. Nick Haley is a colleague of mine, and many of you may know him. He performed a couple of different live animal testing in comparison to in depopulated ranches using a live animal-like sample. And then in actual live animals in an elk study, the PMCA, which you actually heard most of that data earlier, which I didn't know he was going to be presenting, was done for a depopulated herd in blood, using blood. And essentially these two tests and the ones I'm going to talk, the data that I'm going to show you hopefully demonstrate that these two types of amplification tests can increase the sensitivity over conventional antemortem testing. So PMCA, I think we've all seen this slide already, almost the exact same one. So the PRP-C is the sensitive substrate for conversion, is converted by the PRP-SC seed, and through rounds of amplification, sonication, there are more free ends made and more amplifications made, and a logarithmic amplification of the sample is achieved. And this can amplify very, very minute, even single molecules of PRP-SC to a detectable level. So a very, very powerful technique. There are some caveats, there are some challenges with PMCA. The substrate is from transgenic mouse brain, which for me as a biochemist is difficult to obtain, but it's fairly well characterized, the purification, but scaling that type of a sample for testing thousands of samples may be slightly more difficult. There are multiple rounds of PMCA are required for detection where the sample is transferred, typically 2 or 3 times, but there have been studies where 7 rounds of PMCA have been used, and that becomes a lot of challenging handwork. For the blood test, there was a number of purification procedures that were required to get this test to achieve the ultimate sensitivity, and this technique is exquisitely powerful in detecting a molecule of PRP-SC. However, it is very laborious with multiple centrifugation steps and then a Western blot readout where these samples are handled quite a bit, and it takes a number of days to prepare these samples to prepare and then test these samples. So I think some of the, I was going to take this slide out, but I loaded it before he gave the presentation. So this gives the sensitivity, which was previously reported earlier today for PMCA with near 100% sensitivity for animals with brain and lymph node positive samples, and then about a 50% detection rate in brain negative, lymph node positive early symptom disease animals. And this is pretty much parallels what we see in RT-Quic. However, this is for GG animals. I'm going to show you some data that we've done with RT-Quic that is on GS animals and some of the resistant genotypes. So the essential kind of the summary for PMCA, if one were to use this as an antemortem test, is that it has the best sensitivity for prion detection, hands down. If you want to know if a sample is positive or not, this is the sample to use if you think if you want the best sensitivity. It works on almost anything. I've never seen something that you can't find positive using PMCA. It's not a technique that I use, but in the literature there's so many samples, so many different types of samples that have been used with excellent sensitivity. It can be semi-quantitative depending on when the round becomes positive. You can determine how positive a sample is. There are some drawbacks. It's time intensive, and for me, it's not really thought of as a high throughput assay where you could use liquid handling robots and go through lots and lots of samples, thousands of samples in a week. And it is, I think, fairly expensive since it requires a transgenic mouse colony. I think there is some very positive aspects of PMCA. It excels at the best level of detection. However, it may not be the best technique for testing thousands of samples. So the other technique that I've personally used is RT-Quic, and it is analogous to PMCA in that it replicates the aggregated form of PRP SC, except that it uses as a substrate bacterially produced recombinant protein, which can be produced at high levels. And the test happens in a little bit different manner. So the replication and amplification is similar, where the fragmentation is important. However, instead of a Western blot readout, we use a fluorescent dye called thioflavin T, which fluoresces when bound to the amyloid filaments. And so this makes it a real-time fluorescence assay where we can go from zero fluorescence to a high level of fluorescence, which indicates that reaction is positive instead of doing a Western blot readout, which is handy. Some of the other aspects of RT-Quic is it happens in these 96-well plates, and it's a fairly fast reaction time of 24 to 60 hours, depending on what you're trying to detect. Typically, samples that are pretty positive are a 24-hour reaction cycle, like the remote tissue is something we usually use at a 24-hour cycle for. 60 hours is for things like urine or saliva, where the detection levels are really, really low, or feces. The data are in real time. And the reaction setup is similar to RT-PCR, where you can just add samples to the wells of a plate and then put it in the machine and walk away, and it's done. One of the other aspects, actually I forgot to mention this about PMCA. So RT-Quic it uses a serine hamster protein, which is not deer protein. However, it's very, very sensitive to the conversion of CWD, whereas with PMCA, the substrate has to match the seed. However, you can tell strain differences with PMCA. So if you were investigating strains from different areas with PMCA, you could differentiate that with PMCA, since it more accurately represents the disease process. RT-Quic is kind of like it does replicate the protein, the seed, CWD seed, but it doesn't do that in a manner that recapitulates the disease conformer. So it's more of just a detection assay. It's also potentially more prone to spontaneous conversion, where the substrate can convert on its own to a thioflavin T positive state. This isn't something that happens in like every single well in a test. It typically is at a specific rate. So if you test 4 replicates, the chance of one of those replicates being positive is about 1 or 2%. So, and then retesting could sort out those differences, because that's a spontaneous process. It's also semi-quantitative and is, I said that, performed in 96-well plate. So the studies I want to talk to you today about for RT-Quic are 2 different ones, 3 different studies done in elk and deer. These were performed, the testing for the first 2 were performed by Nick Haley at Kansas State. The second one was at Elk Ranch that the samples were sent to CSU, and we did those tests. So the kind of the take-home message here is there was a really good correlation with IHC of remote testing, and in both studies there were increased sample detection with RT-Quic. So 83 positive samples were detected by IHC, and 131 were detected by RT-Quic, which is a 36% increase of detecting the positive. So if you're just looking at this population, and wanted to weed out CWD positives, you would get 36% more using the amplification test. And another test with deer, which I'm going to go into a little bit more detail, we see about the same amount of increase, and this included a number of GG and GS animals. So this is the population breakdown for that deer population that was tested by IHC in retropharyngeal lymph node, OBEX, and remote, and also the remote was tested by RT-Quic. And with the 96 GG animals, we saw greater than 100% detection in the retro and OBEX positive animals. So it detected some of the animals that were lymph node positive only in this case. In the GS animals, which are partially resistant to CWD, there was 79% detection, and in the handful of SS animals, there was a 31% detection, but only in the lymph node only positive samples, because none of those animals were positive in the OBEX to begin with, right? And so for the, in the cases of the GG animals, there were 20% more found with the antemortem test compared to the antemortem test with RT-Quic, and 54% more found in the GS sampling. And this reflects that RT-Quic is more sensitive and more likely to detect the lymph node only positive animals, and the GS animals had a higher representation, or the population had a higher percentage of lymph node only positive animals. So the animals that are more resistant to CWD with a longer disease course have a longer phase where they're not OBEX positive. So therefore, using a sensitive test like this may be important to determine animals in a resistant state that have CWD in the population. And I mentioned that for the SS, there were 0 detected by IHC and 6 detected by RT-Quic in that group. So there's 100% more detected than IHC. So in these deer, as I was saying, the presence, the percentage of animals that were OBEX and lymph node positive decreased as you increased resistance. So a GS animal, there are more lymph node only animals, and in SS deer, there were only lymph node only samples. So since this longer latent phase in these resistant animals requires a higher sensitivity to detect the positives, using an amplification test increased those numbers over what would be achieved with just using IHC on those same samples. So another study I'd like to quickly tell you about is a serial sampling that we did at CSU with GG and GS animals, and these are inoculated with very low dose of CWD, and they were monitored every 3 months by tonsil and rectal biopsy, and those biopsies were tested by IHC and RT-Quic by 2 different methods. And so this is a summary of one of the animals, the data that we collected. So at 6 months, both samples were IHC negative, and both samples were also RT-Quic negative. And at 9 months, we see, again, both samples are IHC negative. However, the tonsil is RT-Quic positive here. So these are 8 out of 8 replicates at about 10 hours positive. And the remote is negative here, and we start to see a little bit of activity in this remote sample. However, that one was not statistically positive. The next sampling period at 12 months, we see IHC positivity in the tonsil, and RT-Quic remains positive. And then we see remote positive, but only in RT-Quic, and then the story progresses until everything is positive. So, and this was a common thing we were seeing as we were monitoring these deer. So we started to kind of go through and make sure that we tested all of these samples with both techniques. And we've kind of relied on the lab of detecting CWD using RT-Quic and then confirming it by IHC, since RT-Quic was a little bit earlier in detection. So this is a graphical representation of 4 different deer. These are GS on the bottom and GG on the top. And you'll notice that the GG deer get sick a little bit faster than the GS deer. And the other thing I'd like to point out is that, so it's kind of hard to read all this stuff, but in most cases, you'll see a tonsil RT-Quic positive, remote RT-Quic positive, and then eventually positive by IHC. So that was consistent for every animal we tested. They were either tested concurrently positive by RT-Quic and IHC, or was RT-Quic positive early and then progressed to IHC. So RT-Quic wasn't measuring a non-specific CWD activity. It was just detecting disease a little bit earlier. The other thing we saw is that in samples where we expected an IHC positive here, but the sample had no follicles, RT-Quic still detected that sample as positive, which is important because this could be 5 to 10% of a sampling depending on the quality of the biopsies. And if you're missing that many samples just by not having a diagnostic biopsy, it would be helpful to have another test which you could use to determine if that sample had CWD. The other main thing we saw with this sampling of the serial sampling of animals is animals that are heterozygous at position 96 were generally detected earlier than the GG animals based on RT-Quic testing. So like I said with the other animals, since these are in a latent period longer, it may be more important to use amplification test to find that early disease point faster. So the summary of this current study it's not published yet, but we're in the process of submitting it, is 11 of the 16 deer were RT-Quic positive before they were IHC positive. So that was, and that was consistent from what we've seen before. 18 of 18 samples that we expected to be IHC positive that had no follicles tested positive by RT-Quic, and this was really consistent. If we missed one late in disease, it was always RT-Quic positive. And I should mention, the biopsies are taken at the same time, not from the same place. We obviously don't know if the RT-Quic sample had follicles in it or not, but the likelihood is fairly high that if the sample is split in half for testing, that there's no follicles in either one, but we can't know for sure because we homogenized it, right? And all 75 samples that tested IHC positive were also positive by RT-Quic. So we have pretty high confidence that if a sample is going to be positive by IHC, it's also going to test positive by RT-Quic. So you don't really miss samples in that respect. And it's a possibility. We still need to do more work. Finding these resistant animals is kind of hard. There's not a lot of samples available for them, but with the limited data we have, it appears as though we get earlier detection of resistant animals using the amplification technique versus immunohistochemistry. So that kind of breakdown is how this might be happening in a group of animals, deer or elk. I know these are elk. Biochemist, I could still figure that one out, but that's about it. So in a population where you have a certain percentage of IHC positive animals that would be detected, you could potentially get a host more of them using the more amplification assays. And like I said, you could use PMCA for this test, or you could use RT-Quic for this test. It just depends on how many samples you want to be testing. I think they would both be just as effective. And in a resistant animal state, if you used IHC only, you may not get very many positives. But using the amplification test, you may actually be able to detect more in that population. And that's still something, like I said, I'd like to work out a little bit further, but getting my hands on those samples has been a little bit difficult. So the main challenges with antemortem testing are the postmortem test for the retropharyngeal lymph node is more sensitive than anything we've been able to use on live animals. So the live animal test is good for sorting out positives, but you're going to miss animals. So you really need to serially test the population over the years if it's in a captive setting to remove all the positives, or if it's in a wild setting, you just have to cut a model in the difference of how many positives you would be getting with either technique. The other thing that I struggle with is how could you ever determine if animals were safe to move or with a high likelihood that those animals don't have CWD. And I think consecutive yearly tests would help to narrow that down. And I think at some place you could get to a point where you would consider those animals safe if they're in some CWD-free environment. And this may be a longer period for resistant animals. I don't know how many years you need to test those animals consecutively for them to be definitively negative. But that's, like I said, I think we're getting towards a better understanding of that, but I'm not sure if we're there quite yet. I don't know how to determine if animals are negative using these tests. So in essence, to sum things up, PMCA and RT-Quic are next generation assays to increase sensitivity for antemortem detection. Amplification methods may be key for detecting CWD in resistant deer or elk that are in an earlier state of disease longer. PMCA is the most sensitive and specific test for CWD that we know of. RT-Quic is fast and it's scalable. It has a higher percentage of false positive reactions, so there's a trade-off with those two tests. However, I think they both work really well for this. But however, IHC detection for live animals is well established and it's widely available, and you're not going to have a false positive problem with that test either. But the trade-off there is it may not be as sensitive as these amplification tests. So that's all I have for you today. I hope we have some good questions in the session tomorrow. And I'd like to thank Ed Hoover, who is an advisor of mine, and Nick Haley, who's a collaborator and provided some of these samples. And we've worked on these tests together, as well as some of the other people in the Hoover lab who've done these tests over the last few years. Thank you.